Hi, we are going to talk about lipids in the GI tract, how they are digested and absorbed. There's a few different types of them, including triglycerides, steroids, and phospholipids. We are mainly going to focus on lipids that are phospholipids and fats, phospholipid fats, and triglycerides. Lipids are distinct in their chemical makeup in that they have hydrophobic qualities mostly, but a little bit of hydrophilic characteristics about them. They're composed of repeating units of fatty acids we see right here. Triglycerides in particular have a glycerol that makes them a triglyceride molecule, whereas phospholipids, as we see here, of the cell membrane barrier, have hydrophobic tails with hydrophilic heads, but they are constituted of fatty acids, a glycerol, and a phosphate group, phospholipid head. So this largely hydrophobic quality about them, triglycerides in particular, makes them struggle more to get across the cell membrane than proteins and carbs being absorbed. So here we begin. Digestion of lipids actually starts in the mouth. Sublingual glands, other salivary glands produce saliva that has amylase, electrolytes, mucus, but also lingual lipase. And lingual lipase is what we see, and it helps to soften the lipids so that lipids can then be turned into part of the bolus of the food brought down the esophagus and into the cardial orifice of the stomach. So majority of lipids are not broken down in the stomach. About 10 to 15% of lipids are when food is being digested. The bolus will make its way down through the sphincter of the stomach, where it's met by stomach acid and an enzyme, gastric lipase. And gastric lipase is produced through these gastric glands here that we can see. The gastric glands are made up of a bunch of different cells, G cells, parietal cells, mucosa cells at the end, but it is the chief cells of the gastric glands that will, in the tissue, that will release gastric lipase so that it can begin processing the lipids through releasing enzymes and stomach acid. And then after this happens, stomach will also go through a mechanical churning that will also further break down the lipids so that it can be prepared into chyme for the small intestine. So the bolus has now been made into chyme, and it treads on into the small intestine. And so the remainder of the fat is going to be processed through a chemical process of emulsification, which we see here. And it will be turned into medium fatty acids, small fatty acids, or long chain fatty acids, depending on what type of fat is being consumed. So bile that is stored in the gallbladder made by the liver is released into the small intestine, and it's going to coat the global droplets with emulsification droplets, and it's going to also work in tandem with this lipase that we see here, gastric lipase. These guys coat the triglycerides and start breaking them down into mono and diglycerides and fatty acids. So just really simple molecules. And this is really the process where lipids broken down now are being absorbed There are two different pathways that will be taken depending on what is a medium fatty acid chain or a long fatty acid chain. Medium chain fatty acids are more directly absorbed by the micelle, you see here, that pick up the fatty acids, cholesterol, and monoglycerides all together. But however, long chain fatty acids and monosaccharides are resynthesized into triglycerides, and those are called chylomicrons. So this journey takes the long chain fatty acids from micelles, but forms them into chylomicrons, and then it makes its way into the lacteal or the lymphatic capillaries to go through into the lymph. Lastly, we can talk about the hormonal release and facilitation of lipid digestion, gastrin secretion, and CCK we see here, or cholecystokinin, help to provide a flow of lipid and absorption through the GI tract. We see that gastrin is released initially. That's with the stimulus of food in the lower stomach. While it helps to stimulate the gastric glands to release enzymes and stomach acid that aid in not only stomach motility, but also just in stomach acid production. And then once food is starting to churn, secretin is working to also lower food into the duodenum, this area. And it's working to increase intestinal enzymes and in tandem with CCK to make the small intestine start to completely break down the chyme here. And so CCK's job is to contract the small intestine and the gallbladder. These contractions will occur and it will release bile from the gallbladder, where it can then all entirely be absorbed.